Good morning, Facebook community, and thank you for joining UCI Mind Live today. This is our first live Q&A episode since the stay-at-home orders, and so we hope that this video finds you safe and well during this challenging time. I'm Chelsea Cox, Associate Director of Education for UCI Mind, and host of this monthly Facebook Live Q&A series, where live viewers have the opportunity to ask questions of our nation's leading experts in Alzheimer's disease and related dementia research here at UC Irvine. We tweaked our topic a little bit for today to address the current state of clinical trials during the COVID-19 pandemic. And joining me to speak on this topic is UC Irvine's Chair of Statistics and Professor of Public Health and Epidemiology, Dr. Daniel Gillen. Welcome, Dr. Gillen.
Thank you, Chelsea. Thanks for having me, and hope that everybody out there is safe and well.
Yeah, so I know that with these safer at home orders and our kiddos at home, we're all learning to balance our schedules in creative ways. So I really appreciate you taking the time to join us and log in today. Now, Dr. Gillen received his PhD in biostatistics from the University of Washington, and he leads the Data Management and Statistics Core of UCI Mind's Alzheimer's Disease Research Center, which is one of only 30 such NIH-designated centers across the country. He is a gifted trialist and widely recognized for his work in the design and analysis of clinical trials for Alzheimer's disease and other health conditions. Dr. Gillen is an elected fellow of the American Statistical Association, and he serves on numerous data safety and monitoring boards evaluating the safety of ongoing clinical trials nationally and internationally. Dr. Gillen has also participated on multiple FDA advisory panels that are instrumental in deciding which drugs get approved and which do not. So, Dr. Gillen, thanks for joining us, and thanks to all of you who are attending live right now. I encourage you to participate in this live Q&A session by typing your questions for Dr. Gillen into the comments box. Please do note that whatever you post on Facebook will be linked to your name and visible to others, so do post responsibly. And we do appreciate if you can keep your questions more general rather than personal and specific. So to get us started, Dr. Gillen, I have a few questions for you. And first is to just kind of basically go over what exactly is a clinical trial, and if you could tell us about the different phases required in a clinical for FDA approval of new drugs.
Sure, thanks, Chelsea. So, broadly speaking, a clinical trial in its broadest definition is a designed experiment to test or assess a new intervention inside of a human population, generally for a particular disease area. We can think about clinical trials in the context or the global context of clinical research, which is defined by research inside of human populations. And clinical trials, if you will, you could think about partitioning that space of clinical research to designed experiments, i.e. clinical trials, and observational studies, which are not designed or do not intervene on the population, but actually collect data on the existing population, hence to observe it, and then try and draw or estimate associations with inside of the population. So clinical trials, again, what makes them stand apart in this realm, this broader realm of clinical research, is that they are designed experiments. In which case that we control or intervene on the population through design. And so we generally think of trials and describe them in terms of the phases as well as their scientific goal. And what I mean by the phases is that when you think about drug development and the drug development process that we have 4 primary phases of clinical trials. So the first would be that you have what's called phase 1, and these are generally smaller studies that are looking at, particularly if you're looking at a biologic or some sort of an intervention that is a biological component, these would be what would be called dosing trials. So the goal of a phase 1 study is to determine, if you will, something we call the maximal tolerable dose within individuals. So we are generally looking at starting in people at very low doses and then ranging them up a little bit, thinking that the more of an intervention or a drug that we can get into someone, the stronger that it will work. We call that a monotonic dose response. But of course, we don't want to give so much of a particular therapy that you have adverse events occurring at a high event rate in people. So that's the target goal, if you will, of a phase 1 study, is really to assess safety and determine what we would again call an MTD, or a maximal tolerable dose. Those are usually very select patients. They're kind of in the neighborhood generally of 15 to 30 individuals is kind of the sample size that we would have for phase 1 studies. And once an MTD is determined from a phase 1 study, you move on to what's called phase 2. And these are really kind of proof of concept types of studies where you look for hints of efficacy. In other words, is the intervention looking to act like you want it to in terms of the outcomes of interest? So if you were looking at, for example, mortality, is it decreasing mortality, or are there hints that it's decreasing mortality? Sometimes we'll look at simple biomarkers, for example, are we lowering cholesterol if it's a cardiovascular disease setting? Or if it's an AD trial, maybe are we lowering amyloid beta, for example, or tau levels in a patient? And so that would be a phase 2 study. And again, it's not meant to particularly prove efficacy. It's more so to give us, I would consider them to be screening trials to say, are there hints that a new therapy is likely to work if we start to test it in a larger population? So those studies can run anywhere from, say, I've worked on phase 2 studies as small as 20 individuals, all the way up to, in fact, as large, depending upon the disease, as 1,000 individuals. And so phase 2 studies can be quite large, but the goal is to use them to screen, to move on to then what we would call a phase 3 clinical trial. And that's what most people think about as clinical trials, because that's what they hear about in the news. Phase 3 studies are kind of your penultimate step that you would have in the trial process before approval from regulatory agencies, for example, like the FDA in the United States, or the EMA, the European Medical Association in Europe, as an example. And so phase 3 studies then are much larger in size. Generally speaking, they're going to range anywhere from, say, 100. There have been phase 3 studies all the way up to 20,000 plus individuals, like the Women's Health Initiative, which was looking at hormone replacement therapy and looking at cardiovascular and breast cancer outcomes. So a very, very large phase 3 study there. But that phase 3 study, again, is really looking to prove or to disregard, if you will, a new intervention that we're considering. And that would be kind of our final step. Now, the FDA regulatory process customarily actually requires results from 2 consistent and independent phase 3 clinical trials. For certain drug areas, for example, AD, where there are chronic unmet needs, then we have what's called an accelerated approval process, where approval can be granted on a single phase 3 trial oftentimes. And I mentioned a fourth phase. Fourth phase is really post-marketing, is what we would call it. So this will be considered an active surveillance study. So, the fact of the matter is that when we develop a new drug, we have evidence on a smaller sample of patients that are generally going to be using that in the population, of course. And so what we use for phase 4 are generally looking to see if there are long-term safety effects that we can pick up in an active surveillance setting. So, for example, collecting adverse events on patients that are using the drug, or maybe looking at longer-term outcomes, for example, like progression to AD, that would take a very long time to look at inside of a study if you began with individuals that were cognitively normal or maybe even mild cognitive impairment. Whereas in a phase 4 study, you might be able to follow those individuals out much longer and see more evidence of this acting in the population. The really big chasm there, though, between phase 3 and phase 4, though, of course, is that in phase 3, we have this design setting where we have randomization and we have control to reduce bias in our outcomes. Phase 4 means that a drug has been approved and it's being prescribed, for example, to the general population. So this is no longer a controlled experiment in the sense that we have comparison groups that we know are fairly balanced with respect to other factors that might impact the outcome. So it's really difficult to infer any kind of causation from something like a phase 4 study. That inferring really comes at the phase 3 setting. Phase 4 is meant to kind of look at long-term outcomes, though, to see if there is other evidence that either on the safety side or on the efficacy side that may be occurring. So, one of the things that people often ask is, well, why is a statistician here in talking about clinical trials? This seems like it should be done by trialists being physicians and clinicians and research scientists that are in the lab, for example. The reason why is because that we have variation in responses. No intervention, essentially no intervention that we have works in every individual, and certainly does not work equally well in every individual. So we call that variation in a response. And that's my job as a statistician, truly, is to quantify variation, to determine if things on average, say, for example, work in the population. And the other aspect is to reduce bias. In other words, are the effects that we're seeing really being driven by a third factor, or are they truly something where we can get closer to inferring cause and effect of a particular intervention? And again, that's really that's the foundations of statistics, is that type of thinking, and hence the reason why a statistician would be here talking about this in study design today for you all.
Why are these various phases of clinical trials necessary to find new treatments for Alzheimer's disease and other health conditions?
So really, it comes down to largely ethics and efficiency. And those two things are actually combined together, I will argue. Certainly if you did not know what a correct dosing level is or had any empirical information on what a correct dosing level is, you would not want to be just experimenting on large numbers of individuals in a phase 3 study. So that really is the rationale behind a phase 1 trial, is to learn that. What is a reasonably safe dose that we feel comfortable experimenting on large individuals with? Phase 2 studies are really the ones that get this question. It's like you have this thing that's a little bit in between, right? I mean, so you have the dose, and you're not really going to get a big enough sample size of individuals to prove necessarily that a drug works. So why are you doing this? Well, the reason why is because you can think about it as like triaging. So again, I mentioned the idea of screening trials. Phase 2 studies really save us a lot at the end of the day, both in terms of numbers of patients that we're using for participants, resources that we're using in terms of study design and drug development, and time, to be quite honest. And that's because phase 2 studies, while they have smaller numbers of individuals, if you see no activity inside of the human in those phase 2 studies, it's unlikely that you want to be going on and experimenting in a much larger population of individuals. So they are really an efficacious aspect. In phase 3, again, it kind of speaks for itself. That's where you want to basically have enough precision in your estimated intervention effect in order to, with some confidence, say this drug actually works. We as public health practitioners, I particularly think of my job as trying to increase the prevalence of truly effective therapies and drugs in the population. That's what my goal is. That's what my life's work is in terms of the theory of statistics and the application of statistics through clinical trial design. Phase 3 studies, adequately powered and well-controlled phase 3 studies, allow us to do that. They are our gold standard, in fact, for establishing whether an intervention truly works and is efficacious, we would say. In other words, have efficacy. And then finally, phase 4, as I said, because we can't run, and particularly you see this these days, right, with the current pandemic, the COVID pandemic, we're not going to be able to run vaccine trials or even intervention trials for an infinite period of time. Those are going to have to shut down at some time in order to become usable for the rest of the population. That's, again, an ethical consideration. It's also a logistical consideration as well. But phase 4 studies are really necessary because we do want to look at things long term. We have particular interventions that we may have studied for 3 to 4 years in a controlled setting, but people are going to take them for 10, 20, 30, 40 years in their lifetimes. And so we want to know what happens with longer and longer exposure, whether it be on the safety or the efficacy side. So there's very good rationale for all 4 of these phases that we have.
You touched on the COVID-19 pandemic briefly, but I just wanted to ask, during an urgent public health crisis like this one that we're currently experiencing, are all of these phases and clinical trials still necessary?
So certainly clinical trials in general are still absolutely critical and necessary. Sound clinical trial design is just as important during a pandemic as it is at any other time. It's imperative that we develop and we provide therapies that are known to work, and that in the way that we intend truly, and that are safe. So you'll hear people say we want to verify that something is safe and efficacious. So there are individuals that might say, oh yeah, but we're in the middle of a pandemic. We should rush to something to put therapies out there that maybe we have a case study or we have anecdotal evidence on. And so why not give people the opportunity to simply use that? Well, there are a couple of reasons why you want to think about that not being a great idea. I mean, the first is to think about what are the possibilities for individuals that might be taking this new drug that doesn't work. Well, first of all, they're not going to be taking something else that might be working, right? So in other words, you're eliminating other treatment options by doing this. And you may be doing that without lack of evidence that something actually works. And so at best, they're utilizing an intervention that they believe to work, but possibly doesn't, and precludes them from using something else, and maybe gives them a false sense of security, in fact, putting themselves at higher risk. The at worst side of this, though, is that almost all interventions have some potential for harmful adverse effects. When you're putting something into the human system, there is a potential for adverse events to be occurring, safety signals. And so in the worst case scenario, you may be putting things out that not only don't work, but may be doing more harm than good. And so in order to avoid that, we do need well-designed trials. And some would say, yeah, but maybe you have observational data. Well, observational data, the problem with it is something we in statistics call, in epidemiology, we call confounding. That, the associations that you're seeing, you might have a group of individuals that look like they do better on a particular treatment who have been diagnosed with COVID-19. But maybe those individuals that were receiving the treatment, for example, tended to be younger in age than other individuals. And so that's why they look like they were doing better. Or maybe they had a particular genetic modification that was leading them to do better than others, for whatever reason. If there's an imbalance in those aspects, then the association that you're seeing is really not the true one that you would estimate and what you would expect to see if you had used that drug randomly in the population. And so we do, in fact, still need clinical trials in this setting. We need to have good, strong evidence about safety and efficacy before we releasing things to the public, because we can't really roll that horse back into the barn once it's been gone. And there are examples of this. We are fast tracking things. We might not need all phases in some settings. So many of you and the viewers that are out there have probably been reading about remdesivir, right? And so this is an antiviral drug that basically blocks mutations. It was originally developed for Ebola, actually. So we fast-tracked very quickly a phase 3 trial in individuals to see if remdesivir lowers on average the time to recovery among COVID patients. So patients were randomized to remdesivir or placebo. And you can see that the results from that, this is one of the quickest occurring trials in history. And that's simply because of the pandemic that's happening right now. But it was done correctly. And we see that on average, we have the time to recovery about 11 days in patients treated with remdesivir versus approximately 15 days in patients treated with placebo. And that was statistically significant. And so this won't follow the usual FDA kind of process in the sense that can oftentimes take up to a year and a half for, in our usual settings. This is going to be a fast track setting. And I would not be surprised if the FDA comes out very soon with some sort of an emergency use verification on this. And there's talk about it already today, in fact. So those data are going to have to be vetted, and they will be vetted more and more. And I think we understand a need for urgency, but I don't also think that we want to forego the scientific principles that lead us to safe and efficacious therapies.
Now, if you're live with us right now and just joining us, just a reminder, you can type your questions for Dr. Gillen into the comments box. And we do have a question here from a viewer. In the news, we're seeing a lot of work toward a vaccine for COVID-19. any updates on a vaccine delivery date, or how long might it take to develop, test, and approve a new vaccine like this?
Yeah, I mean, I think it's an excellent question, and and truly, when we think about shelter in place and we think about the comings of second waves, the true, obviously, big milestone here would be a vaccine, right? And there are many targets that we're considering. Essentially, the way that we're going about those things is looking at the antibodies. Those give us the targets and say if we can develop those antibodies initially, then we may be able to block the virus and hence a vaccine coming through. The reality of the situation is we don't have those we have many target candidates, but they haven't moved on to later phases of experimentation just yet. And, they will be fast tracked. And the likely scenario is that early 2021 is probably when we could foresee maybe a vaccine. And that's if many things fall into place. We move forward with a particular target vaccine. Another big aspect of this, which is different than the way we do a lot of drug development, is that if we do move forward with something into a later phase study, it's going to be necessary to begin to ramp up or scale up, if you will, the manufacturing process for that proposed vaccine, even probably while that study is going on. Now, that's a risky situation. It's not something that companies or the government usually are doing in cases, because you don't really know the answer yet as to whether this vaccine is going to be efficacious or not. But in order to make it useful and in a timely manner, we're going to need to have it mass produced. And so really there's going to have to be an up-gearing, if you will, of the manufacturing process and a fast tracking of the trial process. And then I would say potentially, 2021, very early 2021 is not out of the realm of possibility. That's my own personal thinking on this. I think Tony Fauci has also mentioned probably around January 2021 is when he thinks is a reasonable, or maybe not reasonable, but an optimistic date for having a first vaccine. And so it is going to take a little bit of time. But I do think that what we've been doing is we have certainly been bending the curve. I think we need to be pretty steadfast about that, because I think that as soon as you release and go back, then it likely is to be a second wave. We don't know how big that second wave will be. But unless you've kind of eradicated the virus within the population, then there's going to be probability for transmission. And as many of us know, transmission rates are exponential, and so they will grow quickly in a network. And so a vaccine is definitely something that is on many of our minds. It's currently on part of the research agenda here at UCI. We have an R&D vaccine center inside of the medical group, and they are currently looking at antibody targets for potential vaccines. But I do think it's going to take still a little bit more time. It's not the answer that a lot of people want to hear, that you're thinking a month or two. Most likely we're looking early next year.
Dr. Gillen, I want to transition briefly in our last few minutes here to Alzheimer's disease clinical trials, since that is the work that we do here at UCI Mind. During the pandemic, with working remotely and safer at home orders, what happens to Alzheimer's clinical trials?
Yeah, it's a great question, Chelsea. And there have been varying opinions about this that have been stated about clinical trials in general, and not just AD. And I will speak AD specific. But when we think clinical trials in general, opinions have ranged from completely shut down things because things are too dangerous to have patients moving on. And the other aspect is, should we continue on, in certain situations? Now, the FDA has given guidance on this, and I think, first and foremost, and this goes for myself as well, we want to ensure safety of trial participants, no matter what. And so things are going to be a little bit outcome dependent. We need to consider the protection of participant safety. We need to consider their welfare and what is best served by them in terms of the study. And you're not going to treat all potential trials the same, I would argue. If you're talking about, for example, late-stage cancer therapies, individuals are going to need to be obtaining medications and treatment therapies in those trials and continuing on. For more benign types of scenarios, you may be able to lax part of the protocol. What I don't think we should do is abandon trials that are currently ongoing in particular, and in our AD trials that we're running here. So at UCI, many of the trials that we're working on, what we've basically been able to do is to say, let's do our very, very best to keep individuals retained inside of our study so that we can continue to follow them for the outcomes of interest and try and adhere as close to the protocols that we've specified as possible. Now, with that comes some modifications. Examples of modifications would be where you might normally have an in-person visit. We'll now modify those. So we might have telephone or video conferencing safety visits or follow-ups. Or if we do need an in-person visit, that might take place at an alternate location. So many of the studies that I'm on that require blood draws to assess safety in individuals, we're doing those blood draws at home now. We're actually visiting patients while they're at home. You know, of course, all of these things disproportionately affect our AD trials because that age population is exactly the population that's highest at risk for morbidity and mortality from COVID-19. So we here are very, very aware of this, acutely aware of it. And we're trying to balance the line, and I think we're doing a very good job of continuing on so that we have valid information in our trials at the end of the day, so we can continue to develop novel and more effective therapies for AD and related disorders, but at the same time, again, protecting the safety of our patients.
Okay. And with these modifications that we're implementing, do you think that the pandemic will set us back at all in discovering new and improved treatments for Alzheimer's?
Yeah, so I think that I'm very optimistic. If you think about this in the long run, I do not think the pandemic is going to have do significant damage to the progress that we're going to make in AD research and the development of new interventions and therapies. You know, as we think about the broader spectrum, we are continuing on our research. Again, we are continuing on with trying to follow these protocols the best we can. You know, there is a short-term impact of these things, you know, so clearly we are running into situations where we're having to deviate somewhat from protocols. For any trials that we're going to start up and running, difficulty is obvious in terms of recruitment, which is always a big struggle in clinical trials to begin with, but those aren't going to begin for a little while. You're not going to be recruiting new patients into studies and having to bring them in for visits, for example, during the pandemic. So those are going to be put on hold for a little bit. But I am very optimistic that in the long run, we will in fact continue to make progress, and you know, we'll consider this, at least in my personal opinion, a bump in the road on the longer journey to developing novel and effective therapies.
Okay. I have another question here from a viewer. Going back to COVID-19 very briefly before we have to wrap up this session. But Dr. Gillen, what are your thoughts about needing herd immunity? And do you think that the shelter in place is preventing that to occur?
So, you know, herd immunity is going to, you know, essentially when people talk about herd immunity, you're talking about greater than 50% or even 60% of the population infected. You know, one of the issues with that, and the reason why, in my opinion, shelter in place is a rational approach to the problem is that, first of all, you have to think about the overload on the medical system and the frontline workers, and whether we have the availability of resources to treat patients if we have large numbers of infections occurring within a short period of time. You know, that's this concept of bending the curve through shelter in place. And so certainly if you get to a level of having such a high infection rate across the population, then yes, you can, if in fact people became immune, which we are not still certain about this, by the way. There is still uncertainty as defined by the CDC and stated by the CDC and the WHO. But if you did have immunity to COVID-19 once contracting it, you know, and you had herd immunity, certainly that is one approach to dealing with the problem, you know, but beyond the resources that's there, you have to look at the excess number of lives and comorbidities that you might be adding on to in that context in order to allow for this to happen. So I do believe that kind of stretching out that curve allows us to better treat not only patients that might be infected in the short term, but gives us, if you will, some time to develop therapies like remdesivir. If you think about that, it's dropping the average time to recovery by approximately 4 days. Individuals that were infected back in February did not have that luxury. They didn't, and those individuals affected in March. So we are now allowing for the development of new therapies that can ultimately, hopefully, reduce morbidity and mortality by doing this as well. So that's my general thoughts on it.
All right. Well, Dr. Gillen, thank you so much. A wealth of knowledge. It's always really an honor to speak with you. So thank you for your time today. And thanks to all of you who are attending live right now and who asked your questions. We do hope that you'll join us for future episodes on the first Friday of each month at 11 o'clock Pacific Standard Time. And next month, June 5th, I will be joined by Dean of the UCI School of Biological Sciences and Director of the UCI Alzheimer's Disease Research Center, Dr. Frank LaFerla. In the meantime, during this time, although we cannot be doing in-person education events, we are regularly updating our blog, which you can find at www.mind.uci.edu/blog. So we'll continue to be in touch via our blog with Alzheimer's disease resources, COVID-19 resources, as well as on our social media accounts. And then lastly, I see that we had a question about whether UCI staff members can participate in clinical trials at UCI. And absolutely, for people who are interested in helping researchers at UCI answer questions in clinical trials or other research studies, UCI has a recruitment registry called the UCI Consent to Contact, or C2C registry. And anyone 18 and older can sign up online at www.c2c.uci.edu. So thanks again very much for attending, and we will see you next month. Bye.